All right, hello everyone, welcome back. Last video, we talked about photosynthesis in our very together page. That's definitely not in pieces. We talked about the light reactions of photosynthesis, this light reaction that we're seeing here, and also like the chloroplast, what photosynthesis is, what we're doing. We are taking light and carbon dioxide and water and we're turning it into, most importantly, glucose and a couple other things, but we don't really care that much about those. So when we went through our light reactions, we made a bunch of really cool stuff. We made ATP here and we made some NADPH, and I told you we were gonna need that to make glucose later. Well, now we are doing that. We are making glucose later. It is later. Welcome. Okay, so today we're gonna talk about the light independent reactions of photosynthesis. So it does not need to happen during light. So obviously these are taking in photons. That requires light. You need the sun for photons, unless you have like an artificial light source. That's fine too. The plants live off of those. But you need, in nature, there aren't many of those. So that happens during the day, and that's light, no matter what it is, light dependent. You need some sort of light source. The Calvin cycle, however, is light independent. Okay, so we're still talking about photosynthesis. Let's make a lovely header. Photosynthesis, part 2. Electric boogaloo. Okay, photosynthesis. So if you haven't seen the video on our light dependent reactions, you should definitely check that out. And after this, we're gonna have one more in our little photosynthesis series, talking about adaptations to photosynthesis. We're gonna talk about C4 and CAM plants, if you've ever heard of those, which if you haven't, that's fine, because that's the whole point of what we're doing. We're talking about the Calvin cycle. So before we were talking about the light dependent reactions, now we are talking about the light independent reactions, or the Calvin cycle. You can call it either. I'm gonna say light, just in case you're confused, independent reactions, because it's not dependent on light. Or I guess it's just one. There's just one. Well, no, there's several reactions actually. I changed my mind. This is happening in a stroma. So before we were looking at the interior, the anatomy of a chloroplast and where our light reactions were happening. So remember, the stroma is this fluid inside the chloroplast, and you also have thylakoid stacks called grana or granum if you only talk about one of them. And when we were doing the light dependent reactions, it was happening like in that thylakoid membrane, including the stroma and the lumen. Right now this is only happening in the stroma. Okay, now let's draw a circle. This is not a circle of like, oh, this is not a very good circle, guys. Hopefully your circle is better than mine. This is not like us drawing a chloroplast. Again, it's just kind of a circle representing this cycle because it's a Calvin cycle, so it's like a circle. And we're gonna divide this into 3 parts. It does not have to be perfectly equally sized, but hopefully something like that. It does not have to be perfect. We are learning biology, not taking art class. We are going to start here with someone called rubip. And you're like, who is rubip? So valid. Rubip is a 5-carbon molecule. So I'm drawing 5 little circles representing 5C. And we're gonna use rubip and other stuff to eventually make our glucose. But it is a long process. So glucose is a 6-carbon molecule. Oh, so you're thinking we add a carbon to rubip and we make glucose. No, it's not that easy, but we are gonna actually add a carbon in the form of CO2. So obviously this is 1C, CO2, 1C. And how are we gonna do that? So we are going to add this carbon dioxide to this rubip molecule using an awesome enzyme, are you ready? Called Rubisco. It sounds like Crisco. Call back. So we have this enzyme that I've drawn like a little Pac-Man called Rubisco. This is ribulose biphosphate, maybe? Something like that. You don't need to know what Rubisco stands for, but what it does is it'll bind our rubip. So draw like most of this area filled in by rubip, and then a little bit of space for your CO2. Because obviously, oh gosh, CO2. Obviously this one is 5 carbons, so it'll be bigger, and CO2 is just 1 carbon. Okay, so Rubisco, which is the most abundant enzyme on Earth, by the way, because it's in all plants, because you need this for photosynthesis. is very important because it's binding. This is our first step of the Calvin cycle. So we have now made a 6-carbon intermediate. So this rubip is bound with this CO2 and it makes a 6-carbon intermediate. So this step, this chunk of our circle, is called carbon fixation. And that's because we are fixing carbon. Pretty self-explanatory. Fixing specifically our carbon dioxide with our rubip. Hooray! Okay. So if you remember from our formula before, we use 6 carbon dioxide molecules in this process. So the reason that we call this the Calvin cycle is because we have to go through this cycle 6 times to get 1 glucose molecule. And I'll explain why that is later, but as we're going through the cycle, just remember that every time I'm saying like there's only 1 carbon dioxide, yeah, there is per turn of this cycle. Okay, so now that we have our 6-carbon intermediate, this 6-carbon intermediate is turned into something we like to call PGA, and it's actually 2 PGA. Because a single PGA molecule is 3 carbons. So with a 6-carbon intermediate, we can make 2 3-carbon PGA molecules. And PGA, if you really want to know, stands for phosphoglyceric acid. Okay, now that we have our 2 PGA, we're going on to our next step, which requires 2 ATP. And you're like, oh my gosh, ATP, that's what we made before in our light reaction. So it makes sense that we're using it now. And we'll turn that into 2A, I don't know why I put a dot there for no reason, 2 ADP, and then 2 inorganic phosphates. So remember, our ATP is our cellular form of energy, and if we break it apart into ADP and an inorganic phosphate, it'll release some energy because ATP is kind of an unstable structure, so letting it kind of break down is going to release energy. And this process will take our 2 PGA into 2,1,3 biphosphoglycerate. Okay, just to be clear, there is 2 of the 1,3 biphosphoglycerate. The 2 is not part of the chemical equation, it's just there are 2 of them. And you don't need to know what that looks like or anything, but there's 3 carbons, which makes sense. It shouldn't be any more carbons because we're not adding any carbon. Here, we're not even adding any chemical to this reaction. We're just using energy, and that's gonna change the shape in some way that you don't have to know. Now, we're gonna do something else. And that's gonna earn this step its name. So we're using our NADPH that we made before. We're using 2 NADPH because there's 2 1,3 biphosphoglycerates. And we're turning that into 2 NAD plus 2 inorganic phosphates. And then, most people won't write this, but it's just kind of nice to keep track of your chemicals, all of the atoms that are happening here. There's also going to be 2 hydrogen ions, 2 protons, because you have 2 hydrogen here and then it'll be 2. So now if you look at each individual NADPH, we have 2 plus charges. So that means that each NADPH we use, we donate 2 electrons to our 1,3 biphosphoglycerate. So that's why this step is called reduction. If you remember your chemistry, this is also just a general basic bio fact. If you know the acronym OIL-RIG, it's for oxidation is losing electrons and reduction is gaining electrons. So our 1,3 biphosphoglycerate gained 2 electrons from each. So since there's 2, that makes sense. And then there's 2 here. So each one gained 2 electrons. It has been reduced. So reduction is reducing. And you can just remind yourself this is giving electrons, which I'm going to just write as E negative, to biphospho. I don't have room here. Glycerate. Cool. Okay, now that we've done that, we make this thing called G3P. And we love G3P because G3P is going to help us make glucose. So 2 G3P molecules can make up 1 glucose. And that's another reaction, but we don't need to talk about that. However, you're like, what is the rest of this circle for? And also, if I get a G3P every time I spin the circle, why do I have to spin it 6 times? Why can't I just spin it twice? Well, it's not that simple. Basically, we need G3P to keep the cycle going. So G3P can either keep going through the cycle or we can use it. So we're gonna have to do the cycle multiple times, not only so we can use the stuff for glucose, but also to replenish the cycle so we can use it later. So 2G3P make up 1 glucose, but we need 4, so 4 are needed to replenish rubip. Remember, this U is not supposed to be capitalized. Pretend it's not. Remember, rubip over here, we need that for the beginning of the cycle, and if we just like use our G3P and take it away, we won't have any rubip left over. So how are we gonna keep going with the cycle? So 2G3P make up glucose, but 4G3P are also needed to be made in this process to replenish our rubip to continue the cycle. So a better way to think of this, the way that I prefer to think of it, is every 3G3P. Okay, ignore my little paper strip, I'm out of whiteout. Okay, every 3G3P made, only 1 leaves. Leaves. Okay. So 1 glucose takes 6 cycles. So you're gonna have to go around the cycle 6 times. All right, so we need to replenish our rubip so we can keep going. So let's take a look back up here. Okay, we have our G3P, so every third time we'll get to take a G3P, but every 2, the 2 out of 3 times, we're gonna have to put the G3P back into the cycle. And if we use some ATP and turn that into another ADP plus an inorganic phosphate. And if you're confused about this notation, don't forget ATP stands for adenosine triphosphate, and this is adenosine diphosphate, so we lost a phosphate, and that's this phosphate, and we put the I to say that it's inorganic, which means that it's not connected to an organic molecule, which is something that contains carbon. Okay, so now we are in the regeneration stage because we're regenerating our rubip. So G3P is made into rubip. Hooray! Oh, also I think it's important to note, okay, so technically we're making 2 G3P. G3P is another 3 carbon molecule. That's why 2 of it make up glucose. So when we're having 2 here, we're having 2 here. And if we take 1 out and put 1 back in the cycle, then we'll have 1 here. But remember, it's every third, like 1 of every 3 G3P is made, so it's not like half and half. But anyway, so yes, this is actually the Calvin cycle. So we start with rubip, our 5 carbon molecule, and we use our Rubisco, the most popular enzyme on Earth, and we make a 3 carbon intermediate, which is turned into PGA, and that is we use some ATP, some energy there, to turn it into 1,3-biphosphoglycerate, and then that's reduced, giving it electrons, turned into G3P. We can take some of that to replenish the cycle. Sometimes we can take it to make glucose, and that's given ATP, and then we have rubip again. Okay, so these are our different stages. We have carbon fixation, the first stage, where we're fixing carbon, reduction, where we are reducing, and then regeneration, where we're regenerating rubip. All right, so that is the basics of the Calvin cycle, but I do really quickly want to talk about something called photorespiration. So photorespiration is a thing that plants do that's actually really bad for the plant. So photorespiration is what's going to lead into our adaptations to photosynthesis topic. Photorespiration is this process where instead of bonding to carbon, Rubisco binds the rubip to oxygen, which actually ends up undoing everything we did in the light reactions of photosynthesis. So Rubisco is just an enzyme. Rubisco doesn't know really what it's doing. It just kind of does stuff. And it can bind rubip to carbon dioxide, but it can also bind it to oxygen, which undoes everything that we did in the light-dependent reactions of photosynthesis. So Rubisco can bind CO2 to rubip, but it can also bind O2. And this wastes our light reaction products. And that is not good for the plant. Here's a little image of what the photorespiration process looks like, and as you can see, we're losing our ATP and NADPH, which is the whole point of the light reactions of photosynthesis, and we're not making glucose, which is what we want. So this photorespiration process happens just like by incident. It just can happen, so sometimes it does. And this can be salvaged. Like when this occurs, it can kind of be salvaged using mitochondria and peroxisomes. So peroxisomes are organelles in plant cells that contain a lot of hydrogen peroxide, or H2O2. You don't need to know too much about them, outside of that, but they do have a really important role in kind of salvaging our photorespiration products. Okay, so photorespiration, why does it happen? It happens just because rubisco, it's a carboxylase, meaning it binds stuff with carbon, but it's also an oxygenase, meaning it binds stuff with oxygen. So it just kind of happens. But this occurs more when there's more oxygen. Obviously, if there's more carbon around, it's going to bind more with the carbon. And if there's more oxygen around, it's going to bind more with the oxygen. Okay, so when I was filming, I didn't adjust the camera so you couldn't see anything I was writing. So I'm writing it again on this cover piece of paper. Okay, so photorespiration, it's common in high oxygen environments. So when do we see high oxygen environments? Well, when it's really hot outside, plants will lose a lot of water through transpiration. So they'll close their stomata, those pores that let in and out gases, because otherwise they'll lose gaseous water. And while that's great for preserving their water, it's a problem because they'll experience a buildup of oxygen and they'll run out of carbon dioxide. If you can remember from our light reactions, we are making oxygen gas. That is a product of photosynthesis, is oxygen gas, and it usually exits through the stomata. So if the stomata are closed, then it's going to build up and we're going to see a lot of photorespiration occurring. So rubisco can bind CO2 to rubib, but it can also bind O2, and this wastes our light reaction products. That's very sad. This process can be salvaged with mitochondria and peroxisomes. This is another thing you really only need to know if you're taking botany, but you definitely need to know it occurs a lot in high oxygen environments. So when it's hot, and we'll see there are two adaptations that plants have to this process to prevent photorespiration from occurring, and those are seen often in hot environments. So they close their stomata to preserve water, and it results in a buildup of oxygen. And also, we can just remind ourselves we need 6 turns of the cycle. So you can also just write like times 6, because 2 G3P make up 1 glucose, but we need to replenish the cycle, so we end up having to take 6 cycles. So that's what I would remember here, is that for every 3 G3P, you get to keep 1, and then you need to put 2 back in to keep the cycle going. So yeah, that is our overview of the Calvin cycle. And now we're going to talk about the adaptations to photosynthesis.